Hello, these are flip notes 0.6 about the parts of an experiment in unit 0. I've kind of been building up to this section because we've been referencing that you can't determine causation until you do an experiment. Well, that's where we are now. So let's review that using the scientific approach involves all of these steps, and they are especially involved when conducting an experiment. We form a theory, or we have observations, and that helps us with a theory. We create a question with a hypothesis. We gather samples, and we'll talk about sampling in the next video. We do our research, data collection, data analysis, and then after we've conducted all of our research and data analysis, we have a peer review so we can publish, and then after we publish, hopefully others will continue in the replication process. So let's spend a little bit more time here on the theories and the hypothesis. So a theory is just the collection of observations, but it still has to be confirmed. A hypothesis is a testable prediction where we can measure the results. So a good hypothesis is something falsifiable. It means that it can be tested and proven wrong. A good hypothesis is also something that we don't necessarily automatically know the answers to.
So I have a sample experiment that I always like to use as a reference for this lesson. So in our experiment, our hypothesis or our question that we're asking is, will girls like Johnny, and Johnny's just our hypothetical person, will girls like Johnny more if he wears Axe body spray? Go back to middle school, think about the scent of Axe body spray floating through the halls. That's what we've got going on here. So Johnny will ask 10 girls for their phone numbers while wearing no Axe body spray. Then he will ask 10 other girls for their phone numbers while wearing Axe body spray. So we've got some different things going on here. Our independent variable is what we can control. It's what we are using and what we're manipulating. So that's the Axe body spray. The dependent variable is our result in the experiment, and that's how much the girls like Johnny. So do they like him because of the body spray or not? We have two conditions. Normally we would call these groups, but because we're only using one person in the experiment, Johnny, we can call them the control condition. If we were having multiple guys, we'd call them the control group, and that's Johnny wearing no Axe body spray. And then our other group or our other condition is Johnny wearing Axe body spray. If we'd had multiple guys, we'd call that the experimental group. The experimental group in the experiment is going to get the independent variable. That's the group that we are going to mess with. All right, but we want to go a little bit further than that. So we want to know exactly how we're doing this experiment. Remember that part of the experiment process is that we want other people to be able to replicate our experiment. So when we're planning our experiment, we need to describe exactly how we did it and exactly how we measured things. So we can explain exactly how we measure something by giving an operational definition. So exactly how we quantify things, and we're clarifying our details. So we're saying for our independent variable, just saying we're using Axe body spray is not enough. So we're going to say before approaching the girls, Johnny will spray the Harmony scented Axe body spray evenly across his body for 3 seconds. So we're saying what kind of spray, and we're saying how much. So we've clarified that now. Then we need to say, well, how are we going to measure how the girls like Johnny? So we said one way was that we're going to ask for phone numbers. Another way to measure like or how much somebody likes something is with a survey. So after the experiment, we'll follow up with the girls and we'll ask them on a scale of 1 to 10, how much did they like Johnny? We don't even necessarily have to ask them about Axe body spray. We're just asking them, did they like Johnny? We're also going to measure exactly how many phone numbers he got, but this is 2020, so girls may not give out their phone numbers. So maybe if they gave out their Snapchat info or their Instagram username or their TikTok address or anything like that, we would count that as them liking Johnny. So we are clarifying how we will measure our dependent variable. We'll get to the other stuff that's on this page a little bit later in the video. So again, with experimental research, we've mentioned this in our other chapter, in our other flipped notes. But this is now the only way where we can actually look for causes. So we're manipulating one variable. And in this case, our one variable that we're measuring is Axe body spray. Is that going to get him or going to make girls like him? So we're really trying to control all the other conditions so we can say definitively Axe body spray helps him to get girls to like her. Or big old fail, Axe body spray does not make girls like him. We want to try to eliminate any other variables except for the independent variable, which is the Axe body spray. So with an experiment, we can detect a cause and effect relationship. Our independent variable again, or the IV, is the variable we're manipulating. Our dependent variable is the result. It's the one that's affected by the manipulation. Sometimes we refer to the IV and the DV as X and Y, with the X being the independent variable and Y being the dependent variable. Again, we said that we have to clarify, how will we measure our dependent variable? What are we doing for our independent variable? And again, ultimately, we want it to be clear enough that if somebody else were doing this research, they could do it the exact same way that we did it. Or if somebody were reading about our research experiment results, they would understand clearly how we did our experiment. So they're used to be able to measure our IV and our DV. Here's another example. Let's say our hypothesis is that studying helps students succeed in school. So our independent variable would be studying, that if you're studying, it's going to help you succeed. And then our dependent variable is your success in school. But how are we going to actually measure that? The operational definition is going to help us figure that out. We could say how many hours we're studying. We could say attending a study group. We could say completing a review sheet or practicing a Quizlet for 15 minutes. So we want to clarify our independent variable of what do we mean by studying. And then how are we actually going to measure success? And we could say the grade on the Chapter 1 test or their GPA at the end of their freshman year. But we want to give an exact way that we're going to quantify our dependent variable. So getting to our groups. So like I said, in our example with the Axe body spray, we used an experimental condition and a control condition because we only wanted to use Johnny in the experiment. But the majority of the time, we're actually going to use groups of people, groups of subjects, groups of plants or animals, but there will be groups. So again, our experimental group is the one that's going to get the independent variable. The control group gets nothing, or they get a placebo, or they get something neutral. And we'll talk about placebos here at the end of this video. Now, how do we pick these groups? We want to randomly assign them to a group. So the only way to really kind of eliminate bias is to not like specifically choose like, oh, I want this person to be in the control group and oh, I want this person to be in the experimental group. But we need to randomly assign them. So once we have our participants, we might literally have a hat and have them draw if they're going to be in the experimental group or the control group. I'm actually going to talk about random assignment and picking out our samples a little bit more in the next flipped notes. We want to manipulate the independent variable for only one group. So again, we're trying to isolate variables. So we can say it was because of our independent variable that there was some kind of change. The resulting differences in the two groups must be due to that independent variable. Here we have in this image another example, the effect of this biofertilizer X, called biofertilizer X, on plant growth. So our experimental group is going to be watered with that special fertilizer. The control group is not going to be watered with the special fertilizer. So then we can control, or we can measure how the plants grow, and we can see, did the experimental group that got the fertilizer, did that plant grow more? Another example, will student test scores be affected by distracting sounds in the testing environment? Our control group is going to be in a normal, quiet testing environment. Our experimental group is going to be in the loud environment. And you could say that our independent variable here is noise, or in this case, it looks like there's part of a marching band. And then our dependent variable would be how well they do on the test or their test scores. Now, so our goal is to eliminate any other possible variables so we can say it was our independent variable that caused the change in behavior or that caused the dependent variable. But in reality, it is really hard to get rid of all other possible variables. I like the meme. One does not simply conduct an experiment without extraneous variables. So an extraneous variable is any variable other than our independent variable that could influence the results of our experiment. It could influence the dependent variable. So there's always lots of potential things that can interfere with an experiment. And as a researcher, they want to try to predict what are these things that can get in the way of my experiment. If we can recognize them during the design process or during the design stage of the experiment, and then we can try to control them. Then we call those extraneous variables controlled variables because we predicted, oh, this factor may get in the way. What can we do to prevent that from interfering with our experiment? But sometimes we don't know about an extraneous variable, that something just pops up randomly, or we just couldn't have predicted that. And then it does interfere. So when we've ended our experiment, we realize there was another variable that got in the way, and we would call that a confounding variable. So a confounding variable was not caught ahead of time, and it may have interfered with our data. And we'll give some examples here in just a second. So if an extraneous variable goes unrecognized, then it becomes a confounded variable. But confounding variables are a type of extraneous variable. So controlled variables and confounding variables are types of extraneous variables. So let's go back to our Axe body spray experiment. So Johnny's going to ask these girls for their phone numbers, and we want to know if it's the Axe body spray that is influencing their decision. But there's a lot of other things that could, in reality, influence their decision about Johnny other than just the Axe body spray. So down here, we have our potential extraneous variables. His personality, what if he walks up to them and these girls are just like, they don't like his vibe, or he says something dumb, or they think he's too aggressive in their approach towards him? What if he's not their type? His physical appearance. What if he's too tall or too short? Or he's not, they don't like his haircut. And there's something about just the way he looks. What if he already has a body odor other than just the Axe body spray? So it's not actually the Axe body spray they don't like, it's the way that he smells. What about the girls? What if the girls don't even like boys? This is 2020. We know that's reality, that maybe he walks up to them and he's not their type. So how can we control for those things? So we see those things happening ahead of time so we can plan for it. So how we will prevent confounding variables. Johnny will be given a standard dialogue. So we can try to make sure, he's going to say the same thing to every single girl. And maybe his personality won't even kind of show through. Maybe we're going to give him a standard outfit that he has to wear. And then his hair is kind of be the same as he approaches all the girls. We'll try to make sure that he showers before he does this. So he's like a neutral smell before he applies the Axe body spray. And then let's pre-screen the girls to maybe make sure that, one, that they're single, they're allowed to date, and that they're heterosexual. So he kind of has an equal chance with all of these girls. So because we were able to predict these, those potential extraneous variables become controlled variables. So we presented them from being confounding variables. But let's say something else happens that we can't predict, that as the researchers, we didn't see coming. So let's say the morning before the experiment, or let's go the night before the experiment, Johnny posts racist remarks on Instagram. And it gets around the school that Johnny is a terrible person. Now, me as the researcher, nobody told me that. I didn't know that. And I allow the experiment to go on. And girls everywhere turn him down. So we finish our experiment, and I'm thinking, wow, turns out Axe body spray, girls really don't like it. And then somebody tells me, did you know Johnny put really racist remarks on Instagram? Nobody wants to talk to him. And I realized that could be why. I don't know if they didn't like him because of his Axe body spray, Or I don't know if those girls saw his post and that's why they don't like him. So now that post that he made and his, kind of what that did to him becomes a confounding variable. And we would say that our experiment has become confounded because I can't separate my data. I don't know if they didn't give him their information or they didn't like him because of the body spray or because of his racist online presence. So when the variables were linked, it was not caught ahead of time and it may have interfered with our data. So when this happens, when we have a confounding variable, we say the experiment has been confounded, that there's been a third variable or maybe even multiple variables, and we cannot distinguish between which one was the result for our dependent variable. So confounding variables are unfortunate. They don't necessarily totally ruin an experiment. In this case, it probably would have. But sometimes it's a small confounding variable, and we wouldn't say that all of our research is useless. But we would need to document it. And as we publish, we would need to say there was a confounding variable that had an effect on our experiment. And that could have caused a change in the dependent variable. So again, when we find a potentially confounding variable ahead of time, it's called a controlled variable. And that makes the logic of our experiment remain intact. Now, sometimes there are other variables that we really just can't control, that they popped up, or even if we did know about them, we couldn't really control them very much. And we call these nuisance variables. So it's another type of extraneous variable that we either can't control or there was just no way of seeing it coming. Usually they're situational. Sometimes it could be something like the participants were taking a test and then a lawnmower was mowing outside the window and that was a distraction. Or maybe they were trying to measure people's responses while watching a movie and the power went out for 5 minutes. So now we don't know if their heart rate went up because of the power outage or because the movie was scary. Sometimes it's noise, sometimes it's an uncomfortable temperature. One of the big ones is the difference in IQ. If we're trying to see, what are some effects on testing, one of the big effects is a person's natural level of intelligence, not necessarily how much they studied. So those are things that can be hard to control for. And when we have a lot of those nuisance variables, if it's something we can't control for at all, that could make it a quasi experiment as well. All right, mentioned earlier that a control group has to get something. They have to get nothing, there's nothing that's happening to them, or they have to have a placebo or something neutral. Because like in a naturalistic observation, if they think they're getting something, their behavior changes when they know they're in that experiment. So people they know they're in an experiment, so their behavior could change. So we have to figure out, is their behavior changing because of our independent variable or their expectations? We're going to watch a video clip in class about placebos, about all the different forms they can take. But for right now, let's just know that a placebo is something fake. Think of it as a fake pill. Usually it comes in the form of a sugar pill, but it could be other things as well. A placebo effect is when somebody takes a placebo, not knowing that it was a placebo, yet they still get better, or there's some sort of change in their behavior. So we're usually talking about with a drug or some kind of treatment here. So a person takes this medicine that's not medicine, but they get better. There's a change in their depression. There's a change in their body weight. Excuse me. But they weren't given an actual medicine. So in this case, it was actually their beliefs that caused a change in the behavior. Now, the significance of this is if we're testing a real drug, a real new medicine, we have to know that the change in that person's behavior because they took the actual medicine is from the drug and not just their beliefs. So there has to be a significantly different change in the in the experimental group's behavior than the control group. And when there is, then we know, okay, it was more than just a placebo effect. It was more than just their beliefs. How do we avoid a placebo effect? There's a couple different ways as a researcher. First of all, in a single blind study, which is kind of the minimum of how we do these studies, the participants don't know whether they're getting a placebo or an actual treatment. They don't know if they're in the experimental group getting an actual drug or if they're in a control group getting a placebo. But that still leaves us open to researcher bias, that me as the researcher, I know who got the medication. So I may see things that I'm expecting. I see a change in their behavior and I think, oh, they're better, they're getting better. But I also know that they took the medicine. So in a true study, and this is what's required by the Food and Drug Administration, they have to do a double blind study. And this is where neither the participants or patients nor the researcher knows who are in the experimental or the control group. And they usually do this by having an assistant who actually conducts that part of the study. So then I, as the researcher, as I'm looking at the data and I'm looking at the results, I don't know who got the medicine and who got the placebo. And I can really, truly analyze their results, and my expectations have no influence on it.